Good afternoon. I'm Mark Arjona, Product Line Manager at Distech. Before getting into my talk, I'd like to thank Interphex and Bioprocess International for hosting the BPI Theater, and more specifically, the single-use track of the theater, giving us the opportunity to present Distech's first offering for the biotech market, the BIOne. Bioprocessing as a whole has fully embraced single-use technologies, the benefits of which are widely recognized in terms of safety, cost, and throughput. My talk today will address how Distech has delivered these benefits to single-use bioprocessing. The BIOne is a single-use bioreactor system. It consists of a patent-pending disposable headplate and liner, which inserts into a traditional bench-scale glass vessel. The single-use liner molds to and mimics the contours of your culture vessel, maintaining the geometry, aspect ratio, and volume of your existing process. The single-use headplate is pre-fitted with weldable tubing, allowing the BIOne to interface with your controller just as your autoclavable stainless steel headplate once did. The plug-and-play nature of the system allows for the conversion to single-use bioprocessing in a matter of seconds, effectively eliminating the time and costs associated with cleaning, autoclaving, and assembling a traditional benchtop bioreactor. All of this is feasible with your existing vessel, controller, probes, and agitator. All materials are USP Class 6 and animal derivative free, ensuring biocompatibility with your process. The high-density polyethylene headplate on the BIOne has all of the same features as an autoclavable headplate. It houses 4 probe ports, 2 of which fit standard 12mm probes with PG 13.5 threads. The DO port contains a polycarbonate down tube with a thin silicon membrane, alleviating the need to autoclave the DO probe. In order to fully exploit the advantages single-use offers, users will want to take advantage of the dedicated optical pH probe port, completely eliminating the need to autoclave the probes. Culture additions are made via weldable C-flex tubing. 0.2 micron vent and inlet filters are in place to provide proper gassing while maintaining sterility. Flute and microsparger configurations are available. Agitation is facilitated via pitch blade impeller in a vortex orientation. The BIOne will come in 4 configurations, 2 and 5 liter capacities with either a flute or microsparger. These initial configurations will address the majority of batch and fed batch applications. In development is a septum port to facilitate small-scale culture additions, as well as various impeller and sparger configurations. Also, future iterations will address microbial fermentation applications. Because of the growing trend towards continuous culture, we're working on an interface for cell retention devices, which would allow users to model their perfusion process in the BIOne. With regard to your current or future application, the BIOne will be able to accommodate customization. The low antioxidant bioprocess film used in the liner is manufactured in the US and is comprised of 3 layers, each serving a specific function. The low-density polyethylene outer layer lends structural integrity and mechanical strength to the culture chamber. Moving inward, the ethylene vinyl alcohol middle layer imparts optimal gas barrier properties to the liner. And finally, the innermost ultra-low-density polyethylene contact layer provides a biologically inert interface for the culture. In the manufacture of bioprocess films, stabilizers are added to protect the material during processing and throughout the shelf life of the material. The low antioxidant content in this critical contact layer minimizes exposure to the degradants, which may be released following sterilization via gamma radiation. BDT-BPP, a degradation product of the polymer stabilizer known as Ergophos 168, has been shown to inhibit cell growth of sensitive CHO cell lines. The BIOne's ultra-low-density polyethylene contact layer has a reduced ergophos content, minimizing the risk of cytotoxicity while maintaining polymeric stability. The thin profile of the film allows the liner to mold and conform to the contours of your glass vessel, maintaining its volume and capacity. Collectively, these features reinforce the BIOne's utility as a process development platform for even the most sensitive cell lines. Leachables and extractables data will be available from our supplier. Here we have a workflow comparison demonstrating the enhanced efficiency the BIOne delivers to upstream process development. This example compares the individual activities and time involved in setting up 24 autoclavable bioreactors versus using the BIOne subsystem. As you can see, the BIOne completely eliminates the cleaning and assembly steps in the workflow, and if users opt for the optional optical pH probe, can eliminate the critical and time-consuming autoclaving step completely. The result? A 48-hour time savings, maximizing efficiency and throughput of your bioreactors. In evaluating the suitability of BIOne as a culture model, we focused on two aspects: process control and cell culture performance in a chemically defined fed batch CHO process. Relative to a glass vessel control, process control was assessed by monitoring the BIOne's ability to maintain temperature and agitation set points, and maintain dissolved oxygen and pH profiles over the course of the run. This study also evaluated CHO cell culture performance and protein production in the BIOne. To minimize variability, identical process parameters and equipment were used for both the BIOne and the glass vessel control. Additionally, normalized Titer results were calculated from total protein produced in the BIOne system and the control once culture viability reached 80%. These figures show that the BIOne subsystem effectively maintained the temperature and agitation set points of 37 degrees and 125 RPM, respectively. These figures depict the dissolved oxygen and pH profiles of the BIOne and glass vessel control. On the left, we see cultures were inoculated at 100% DO and maintained a set point of 50% once oxygen reached this nominal value. Set point for pH was 7.0, with a deadband range of 6.7 to 7.3. The BIOne was able to effectively maintain pH within this range. Similar dissolved oxygen and pH profiles were maintained in the BIOne relative to the glass vessel control. In these chemically defined fed batch CHO cultures, growth profiles looked very similar in the BIOne relative to the glass vessel control. In both cases, peak densities of just under 9 million cells per mil were observed on day 6. Final titer was assessed once cultures reached 80% viability. Final titer in the BIOne was quantified and normalized to the control. The BIOne's resulting titer was comparable to that of the glass vessel control. In summary, we concluded that the BIOne is suitable for mammalian cell growth and recombinant protein production. By using the BIOne, process control capabilities were not affected, and the resulting growth, viability, and protein production were comparable to the glass vessel control. Lastly, the BIOne significantly reduced turnaround time and costs associated with the cleaning, assembly, and autoclaving of the glass vessel control. This study demonstrates suitability of the BIOne as a robust culture model for CHO cell growth and recombinant protein production. In addition to CHO cells, we've observed comparable cell growth in hybridoma, carcinoma, and mouse cell lines in the BIOne relative to its non-disposable counterpart. We're excited to bring Distech's BIOne subsystem to market in summer 2016.